right, folks, we are going to go over this table so that if you still had questions, you can have them answered. The first question or first thing that we're talking about is which type of cell is produced. And so if you would like to tell me what you put there, please raise your hand. I love how brave everyone is in this class. It's dead silence, hey? Okay, fine. Oh, yes, go. Yeah, this is what somatic cells are doing. And on the other side, maybe you wrote gametes, because that's what we're producing. Now, if you read that and thought I was asking, am I making a haploid or a diploid cell? You're not wrong, right? You could have answered. I wasn't super descriptive with my language. But I am asking about the ploidy later on, and so that's where I'll put those answers. Do the homologous chromosomes align? In other words, pair up? Anyone want to tell us yes or no what we should put? Yes, everyone at the same time. Super duper. Okay, fine. In mitosis, no. In meiosis, yes. In mitosis, the homologous chromosomes do not pair up. They're just chromosomes. Yes, I have 2 of every chromosome, but they don't pair up. They don't form tetrads. They do not line up. The location in the body, a few of you asked me about this because I think you were just looking for the right words to describe this. For mitosis, I saw somebody write this, but like really anywhere. Remember I said it could be a muscle cell, bone cell, liver cell, skin cell, right? Basically anywhere you could see mitosis happening. If we're talking meiosis though, very limited region in the body. Only germinal cells do that. And if you gave some specific terminology like the place where sperm and egg are found, or you called them testes and ovaries because you already knew that vocabulary, that would be a reasonable answer to give as well. Now for mitosis right here, prophase, metaphase, prophase, metaphase, anaphase, telophase, cytokinesis, and that is mitosis. That is one division. I started with 1 cell, now I have 2. It divided in half. That is counted as one division. But if you look on the other board, you can see prophase, metaphase, anaphase, telophase 1, and then the same thing repeats. So there are 2 divisions. The next one is really important. Is there DNA replication before each division? Well, because of these handy drawings that are here, the mitosis cell cycle always has interphase. As soon as the cell is done mitosis, it goes back into interphase. Then it can go through mitosis again. So the answer is yes, there should be replication of DNA before each division. That way, I'm always going to have the same number of chromosomes. I will never change the number of chromosomes because I'm always going to copy them, split them in half. Copy them, split them in half. So I'm not going to reduce the number. But for meiosis, I would have to put no. It's true that before meiosis 1, there is replication of DNA. Look at Mrs. Polgar's gorgeous pictures. Here's G1, there's S, where the DNA is being replicated. Then we go through meiosis 1. But there's no interphase in between meiosis 1 and meiosis 2. So I would have to say no, because it doesn't happen every time, only the first time. 2 daughter cells in mitosis, 4 in meiosis. Quite a few of you asked me to repeat what ploidy means. I'm asking how many chromosomes, what's the chromosome number? And you can respond to this with N or 2N, or if you chose to write haploid or diploid, that's fine too. But that is how I would represent the ploidy. I would ask you this. I would use what's the ploidy of the cells, and want you to tell me N or 2N as the response. In mitosis, the daughter cells can continue to divide. As soon as they're done mitosis, they go through interphase and go back through mitosis again. It's a cycle. But in meiosis, no. Once we're finished meiosis, that's the end of the line. And they're going to have to find another gamete. Fertilization will have to happen, and they'll have to make a whole new different kind of cell before any sort of cell division can happen. Is the genetic material identical? Yes and no. Yes, in mitosis, it should be, because all we did was make copies and then split them apart. But in meiosis, please remember, there were several ways that the DNA was different. First of all, we don't have the same number of chromosomes anymore. We split everything in half. We also talked about crossing over, independent assortment. In the rare case of non-disjunction. There are lots of things that could happen in meiosis to cause the genetic material to not be identical. Now, does anyone want to ask anything about this table? So we're going to continue on with the other part of this lesson. It's really still about meiosis, but we are going to specifically talk about how exactly it looks for sperm cells and how exactly it looks for egg cells. We're going to do sperm cells first because it's simple. Now you'll see this word at the top, spermatogenesis, and you'll be like, oh, that sounds fancy. Genesis means the beginning, so spermatogenesis is the beginning of sperm, okay? That's what we're talking about. Spermatogenesis is what we call meiosis, specifically for sperm cells. What we talked about yesterday and this morning, that was just general meiosis, but this is specific to how humans are making sperm cells. We always start out with a spermatogonium. You can see them up here. The spermatogonium, that's our germ cell. The spermatogonium is 2N. Okay, it has 2 of every chromosome. And you might be interested to know that a little bit of mitosis happens here. That spermatogonium will go through mitosis to maintain the stock of spermatogonia. Spermatogonia is the plural of spermatogonium. It's one of those weird word endings. But some of the spermatogonium cells will start going through meiosis. So we'll have a spermatocyte, and you might remember that I said that cyte means cell. So if you see that in a word, we're just trying to represent the word cell. We can call it first order, or we can call it primary. We're trying to get across it's the first one. And if you take a look at the diagram, the primary spermatocyte is the one that is going through meiosis 1. Okay? Meiosis 1 is listed here. I'm not going to repeat what the steps of meiosis 1 are because we did all the details yesterday, but that's what we call it specifically for sperm cells. At the end of meiosis 1, I have 2 daughter cells, and we call them daughter cells even though a male is probably the one doing this process. But we end up with second order or secondary spermatocytes. Now you might remember that at the end of meiosis 1, we changed the ploidy. As soon as telophase 1 was over, we called those cells haploid, or N if we're talking about chromosomes. So these cells are haploid, and it goes through meiosis 2. Now, at the beginning of meiosis 1, we have 2 of every chromosome, and they still have their chromatid attached, their copy. At the beginning of meiosis 2, I only have 1 of each chromosome in each cell, but they still have the sister chromatid attached. Remember that that was a distinctive thing, that the sister chromatids stayed attached through all of meiosis 1. That will leave us with a spermatid. These are our daughter cells. And they, of course, are haploid. And then eventually these spermatids will mature, and you will see they will gain a shape, or they will develop a shape that's more conducive to them moving where they need to move. We're going to talk a bit more about sperm maturation and development later in the course when we talk about reproduction. And so just know that each of those spermatids just needs to develop and mature and acquire some structures to make them mobile, but the number of chromosomes is not changing. Now that's spermatogenesis. Basically, it's meiosis with some different words. Instead of just calling it a cell, we're calling it a spermatocyte. But what is happening here is exactly how I described meiosis yesterday. Does anyone want to ask anything about spermatogenesis? Okay, so we did this one first because it's simpler, right? It's just what we talked about with a bit of different vocabulary. The other side of the story, oogenesis, with two O's at the beginning of the word, it's a fun word. There's more detail. Everything is more complex with females, just like in general, but in particular with reproduction. Unlike spermatogenesis, oogenesis, genesis means the beginning, so it's the beginning of the egg, is asymmetrical. So I'm guessing someone knows what the word symmetrical means, where the two sides are the same, or it's a mirror image, something like that. I don't know what the math definition is. Do you guys do that in math? We have to make like reflections of things on a graph. That sounds mathy. Anyways, this is not symmetrical. And we're specifically talking about cytokinesis, which you already know about. That's the division of the cytoplasm and the contents of the cytoplasm of the cell. Because of course, we've already divided the nucleus during the process of, in this case, meiosis. Now, oogenesis happens in the ovaries. So those are female reproductive structures. You will eventually have to identify ovaries on a picture, but not at this point in time. We always start with a diploid germ cell. It's called an oogonium. And so you can see on the picture on the right, I'm just describing what the picture says. I always try to put a picture and a description with words because some people like the picture more than the words, some people like the words more than the picture. So you can see 2N myogonium. It goes through mitosis, just like with the spermatogonium, to form first order or primary oocytes. You can see on the picture, these are still 2N because it was mitosis, and mitosis does not change chromosome number. Now that happens before a girl is even born. So while a girl, a female, is developing in her mother's womb, this part is already happening. So before a female was even born, this bit of stuff was going on. These oocytes are immobilized in prophase 1 until puberty. So a girl, a female, is essentially born with the primary oocytes that she's going to have. There's a limited supply. It's not the same as with spermatogonia, where the supply is virtually unlimited. So immobilized means stopped. They don't continue. They're pausing here. And they pause here until puberty. And puberty is another thing that we'll talk about a bit later on in the course when we talk about hormones. But in general, there's some hormone signals that tells when to start again. So once hormones are being made in puberty, that's the signal to these primary oocytes to continue their pathway through meiosis. Now, once this hormone signal tells these cells to continue, my primary oocytes are going to go through meiosis 1. And asymmetric cytokinesis will have taken place. So take a look at these two structures. One of them is big, almost the same size as my original oocyte, and the other one is very small. The reason is one of those two cells that I'm producing as a daughter cell in meiosis 1 is getting virtually all of the cytoplasm. The one that receives more of the cytoplasm we call the secondary oocyte. So you can see it right here. It's big. The other one, so the other cell that got made during meiosis 1 is called a polar body. The polar body receives basically no cytoplasm, but it does receive half of the chromosomes. So we've achieved the purpose of meiosis, reduction division, right? We took the chromosomes and we split them in half, but we gave one of the halves all of the cytoplasm and the other none. The polar body is not going to continue with meiosis. Okay, so it's done. It's finished. It's going to go off. It's going to break down, but it is done its pathway through meiosis. Only the secondary oocyte is going to continue. Anyone want to ask anything there? Now, the second order or secondary oocyte will undergo meiosis too, and the cytoplasm is still unevenly distributed. So here's my secondary oocyte. This is the same picture that was on the slide before. I'm still getting a polar body and this other structure. The one that receives the most, that is going to be the ovum, which is the fancy word for egg. Ovum is a word like spermatogonium, where if I want to say there's more than one of them, I could say ova, that would be the plural. Ovum is singular, and you can call it an egg if you want to. I will understand that. But I also want you to recognize that you could see the word ovum. The ovum, again, is immobilized. So there's two spots where stuff waits. The ovum or the egg is immobilized in metaphase 2 until fertilization. So it doesn't even really finish meiosis unless it's fertilized. Okay, so unless a sperm comes along and fertilizes. The reason that one of these cells had to receive more cytoplasm is because of this point here. That ovum, if it's going to get fertilized, is eventually going to become a zygote, and then an embryo, and then a baby. Something needs to support that zygote as it moves along and makes its way to the uterus where it will implant. And so it needs a source of nutrients. It needs a lot of cytoplasm, so lots of metabolic reactions can happen, and we can support the life of this developing cell. So that's why we need asymmetrical cytokinesis. Just like in meiosis 1, the other cell that we would make in meiosis 2, again, is a polar body. Where are we at in time? Now I think we're going to pause here, and we'll all